All right, so I'm going to share a screen for the review. Okay. All right, so here we are. So final exam should be 100 points, about 10% of your final score. Mostly it'll be multiple choice. I won't have fill in the blanks. It'll be some true, false, and then one essay question. Okay, so prepare for the final exam. You can't reread everything, but you could look over chapter summaries. You could check vocabulary. That's a really important thing. And you can look at Moodle to find all those things, all the cheat sheets and things like that. The lectures, that's all there. It's important that whenever you do a final is to recognize that you shouldn't sit in one long session, but frequent short study sessions are probably the most effective way. All right, so our course was divided up into sections. The first section was background to biology. It covered how science works, the focus on evidence and experiments and predictions. Theories are supported by evidence, and so science works on using the currently best explanation, the one that's supported by evidence that predicts new evidence. Then there's a lot of chemistry in biology, sorry, but it's not, okay. I won't ask a lot of basic chemistry, but sometimes there'll be some basic chemistry type questions that require understanding of that. And then this was our first introduction to cells. Cells are one of the foundations of biology. We should know the types of cells, eukaryote and prokaryote cells, and particularly the functions of eukaryotes. Okay. The second section was really on how cells work. Okay, we talked about the chemical reactions of cells, aerobic, anaerobic or fermentation, photosynthesis, making energy from sunlight. Then we talked a little bit about mitosis and meiosis, but I think I might have moved that back, so I can't remember. But okay, then the third section, that was the inheritance section. We talked about Mendel and unraveling the mystery of disappearing traits. And so we talked about dominant and recessive alleles, the difference between what genes an individual has versus their physical traits. Those can be different. And then that was really fully flushed out. Mendel's ideas were brought to fruition with the understanding of DNA, and that happened about 100 years later. And so that DNA was made up of sugar phosphate backbones and then these bases. And these bases form very specific bonds with other bases that hold the two strains together. And so the sequences of any DNA molecule could be unique. And so it formed a code for making proteins. Okay, and so a gene is a code to make a protein, and ribosomes, all cells have ribosomes because they make protein. And then we went into, when we got back from the break, we went into DNA technologies, moving genes from one organism to another, like BT corn or making human insulin in E. coli, and then things like DNA profiling and stuff like that. Okay, then our fourth section was on evolution, very closely related to inheritance, the idea that organisms can change over time because what genes become common change. And so individuals can't evolve, but populations and species can. Okay, and so we talked about the evolution of animals, and we talked about evolution of humans. And then our final section was on ecology, was the study of the interaction of organisms with one another in their environment. Organisms, we talked about adaptation, biomes, climate, and then populations. Okay, so that's the general overview. Okay, then let's go into some specifics. So here's another set of study tips. Spend lots of time, be active, plan your time, and then take breaks. Don't just keep at it. Okay, so the core themes covered in chapter 1, science, evolution and natural selection, molecules, and then cells. Biology is kind of built around those core themes. In chemistry, there were two types of bonds that we primarily dealt with. Ionic bonds, where there's a transfer of electrons to form ions. The charged ions then are attracted to one another, and so they hang around to form crystals. And then in covalent bonds, kind of shown over here with the two hydrogens, that's the sharing of electrons. And so the sharing of electrons means that the electrons orbit two atoms, and that those shared orbitals are what holds the atoms together. And then some atoms have unequal sharing, and so that's polar covalent bonds. Okay. We also talked about specific types of molecules. For us, the most important is DNA. The double helix, where the sugar phosphate backbones are held together by these pairing of complementary bases. Cytosine pairs with guanine, adenine pairs with thymine. And so these are complementary. And then another really key thing was that most molecules that are doing important functions in cells are proteins. And these are made up of 20 different amino acids bonded together with peptide bonds. And they can form very complex three-dimensional shapes due to folding and hydrogen bonds and things like that. And so they can be used for any function. All right, does anybody have any questions? These are things a while back. Okay, so we talked about cells. Living organisms are made of cells. So here's a bunch of cells kind of together. There's a nucleus, cell wall. These are probably plant cells. All living things are made up of cells. Cells give rise to other cells. The only thing that's alive are cells. Life processes occur within cells. This is these little guys. These are prokaryotic cells, and here's a diagram of one prokaryotic cells. These are the simplest kind of cells, bacteria. Bacteria just have one fluid-filled compartment with a cell membrane around that, and then a rigid cell wall. This fluid in here is called cytoplasm. It contains one DNA molecule and ribosomes. Then there's lots of enzymes and stuff like that. But bacteria are generally very small and kind of all similar. Okay, eukaryotic cells are much bigger. They're 10 to 100 times bigger. They may have a cell wall, but they always have a plasma membrane. They too have a fluid-filled inner space called cytoplasm, but then there's lots of organelles inside there. Structures that are like little organs, like there's this guy there. These little organs perform specific jobs. They're often walled off by their own membrane. So you have a nucleus that contains the genetic material walled off by the nuclear membrane. You have the mitochondria that contains the enzymes for aerobic metabolism walled off by its own membrane. This big thing here, this is the endoplasmic reticulum. It contains its own membrane, or sometimes called the endomembrane system, making proteins and fats. Okay, so more complex, much larger, much more DNA. This is our cells, plant cells, fungal cells. If it's an organism that you can see, it's made up of eukaryotic cells. Okay, then we talked about the chemical processes in cells, and so that's enzymes. So enzymes are shown here. They're reusable, usually proteins, sometimes RNA. They bind with a substrate and they put it in a position where it's likely to interact. Okay, the enzyme itself is not changed, just the substrate is changed into product, and so the enzyme can be reused. Okay, our chemical reactions are all controlled by enzymes. All life organisms have chemical reactions controlled by enzymes. And we have lots and lots and lots of enzymes. That's what most of our genes do. Okay. Metabolism, the ability to get energy from food, is based upon many enzymes working together to step by step take apart food molecules. And so here this shows the step-by-step taking apart of food in aerobic metabolism. So it starts with glycolysis, then you have pyruvate oxidation, then the Krebs or citric acid cycle, and electron transport change. So this is like overall like 30 some steps needed to get energy out of glucose completely. breaks it down to carbon dioxide and water, and then we get 36 or so ATP. Another big chemical reaction that doesn't happen in humans, but we depend on that, is photosynthesis. Photosynthesis takes water and carbon dioxide and sunlight, and it makes organic molecules like glucose. So you have the light-dependent reactions that occur because of chlorophyll. These occur in the thylakoids of the chloroplast. And then in the stroma, the fluid around the thylakoids, occur the Calvin cycle, or sometimes they're called the dark reactions. They use the energy and NADPH made in the light reactions to stick hydrogens on the carbon dioxide. And those hydrogens then allow this to be an organic molecule like glucose. And so everything we eat comes from photosynthesis, whether it's a plant or an animal. The animals have just taken the, if it's a cow, the cow is eating grass or something that's been made by photosynthesis. And so we depend on this. Whoops, what happened there? Okay, another really important set of things that occurs in cells is cell reproduction. Okay, cells go through a cycle called the cell cycle that most of the time they spend an interphase where they do their normal growing and functioning, but eventually they decide to reproduce. So they copy their DNA in the S part of interphase, that stands for synthesis. Okay, after the DNA is done copying, they spend a little more time in interphase, but they're doing different things there. They're making proteins needed for cell division. And so then they enter the M phase, where they store their chromosomes into compact little materials and then separate them. Okay, so this is meiosis, a specialized form of cell division, and this has two linked cell divisions along with some recombination that occurs in prophase 1. Where's prophase 1? Here, I forget. Okay, this is meiosis 1 here, and this is meiosis 2. Well, anyway, it's another type of cell division needed for sexual reproduction. This is not the best slide. Oops. Okay. You can make comments if you want. All right. Does anybody have any questions? I'm just kind of, going over things. Anybody? It's like I'm shouting into the void. Okay. All right. Inheritance. Okay. So there were some things that were always understood about inheritance. Offspring generally resemble parents, but the pattern of traits appearing and disappearing was mystifying. And so how did this exactly occur? That was unknown. And particularly that some traits would disappear. Some kids would resemble a grandparent or an aunt or an uncle more than a parent. And so that was difficult. Mendel came out of really agriculture, and in the 17 and 1800s, ag was far in advance of science. They were selectively breeding both animals and plants and to improve the characteristics. And so they would carefully keep track, or at least some of them would carefully keep track of traits. And when they saw a trait they liked, they would try and fix it. And they did this by something called back crossing, by breeding an animal to its relatives. And so Mendel used a test cross, which was similar based on that. And he kind of knew how this worked because he had been raised on a farm. And so he took true breeding varieties of peas, and he crossed these true breeding varieties and then kept track of specific traits. And so he might keep track of something like flower color. And he recognized that individuals received genetic information from each parent, but that not all that genetic information was equal. Sometimes those individuals had genetic information that could remain hidden. And so he referred to these as recessive traits, and traits that showed up all the time he called dominant. And that individuals could carry 2 copies for each trait, 2 copies of genetic information, and that They could have 2 copies of the same allele, so we're true breeding, or copies of different alleles, heterozygotes, where they were not true breeding. And so by keeping track, he recognized that he could produce these 3-to-1 ratios in the F2 generation. And so this was, this allowed him to kind of figure out that alleles remained separate. There wasn't blending, but that alleles sometimes could be hidden, recessive alleles could be hidden by dominant traits. And so this was the beginning of understanding of genetics. And then 100 years later, using information from X-ray diffraction, we figured out the structure of DNA, and we figured out all cells have DNA, and that the genetic code was the same in all cells. It was used to make proteins in a 2-step process, transcription and translation. And 3 bases could be translated into an amino acid. And so this was how proteins were made. And so these codes here's the genetic code. The 3 bases on RNA can be converted to an amino acid. And here is the code. It turns out it's the same in all animals. That was, and not all animals, all plants, even all bacteria. And so you could take a gene from anywhere and put it in anywhere else and get the same protein. Pretty amazing. And so that was what led to the genetics revolution. We could take DNA out of one cell and put it in another cell, and using enzymes, we could put that DNA and make it back together. And then we found things like phages and plasmids that specially carried genes into cells. And so those cells then would produce the proteins. And so we could take a pancreas cell, get a gene for human insulin, and we could put it in a bacteria, and the bacteria would make the insulin. And so you could treat diabetes. Or it could be the gene for growth hormone, and you could collect growth hormone and use it to treat dwarfism. And so there were many, many applications for this in medicine, but then some of the most important ones also became in gene for pest resistance, genes for herbicide resistance that were really valuable in agriculture. Okay. We also recognize that you could identify, make copies of genes that were left in, like at a crime scene, and we can make that unlimited copies. We could find specific sequences. We could determine sequences, and so we could use DNA technology to identify samples from suspects, from victims, to establish paternity, to ID samples that we find in the environment. We can reconstruct the past based on this. And so it was really important advances in lots of different fields. And this process is automated. So like this lady here, she loads these samples in, and then she goes to drink coffee for 3 hours while the machine works, and then she'll read the data. So really great for biologists. Okay, all right, and then this idea that of genes that we could peek into genetic change also is similar to the same story that happened with Darwin. About the same time Mendel was working, Darwin figured out, okay, there has to be genetic change in populations. Again, because agriculture was far in advance, you could see genetic change happening in agriculture, that you could change organisms and those traits were inherited by their offspring. And so he suggested the same thing was going on in natural populations, that they were changing, and this could lead to populations on separate places becoming different, getting different traits, and new species arising. And so the idea of genetic change meant that species weren't fixed, that we should expect extinction, but also new species and new adaptations could evolve. And this kind of explains some of the similarities between existing animals, that they shared a common ancestor, and those ancestors might be far, far back. Okay, and so what was the evidence of this? Well, Darwin, he cited artificial selection as an example, but also there were good examples from fossils. How do you explain the existence of fossils that appear like no living animal. They appear to be half reptile and half bird. This is Archaeopteryx here. And so this kind of explained that. It also explained the biogeography, the distribution of plants and animals across the world, that, okay, that, you know, that they spread, and then those populations that are far apart are going to become different, and they'll develop into different species. And then likewise, the homologies in development and anatomy can be spread this way. And also the molecules, when we started looking at the molecules, we can track how close these organisms are. Okay, and so then we looked at the evidence for evolution of animals, the similarities all animals face, and then the differences. All animals are eukaryotic, multicellular. They share patterns of development. They share genes. They share traits. And so we look at those things, and we can see the main characteristics of the group. The simplest groups have the oldest fossil records usually, and they are the most distant from most other animals. And we can also extend this to humans. Okay, clearly we share traits with animals. Okay, we are clearly mammals. We have all the mammal traits. Hair, live, birth to live young, nourished by a placenta as they're developing, and nourished outside the womb with milk. We are primates with opposable thumbs. We have fingernails. We have eyes and big brains. And there's some fossil intermediates that look a lot like us and behave like us, because we can find evidence of things they were doing. Okay, then our last section was on ecology, the study of connections in nature, the relationship between organisms and other organisms and organisms in their environment. We can work at these different levels, individuals, populations, communities, ecosystems, or whole biomes. Oops, the other way. In the chapter, at the lowest level, when you talk about individual organisms, they must maintain homeostasis, survive in their environment. And so they have adaptations to allow them to survive in that environment. They all deal with these abiotic factors, energy, temperature, etc. And so their responses can be physiological, anatomical, or behavioral. And we also talked about biomes. These are large areas distinguished by their predominant plants and associated with particular climates. So what happened there? I don't know. They're most important things for terrestrial biomes are temperature and precipitation. And so we see those types of things there. We also talked about populations. Populations have characteristics like age structures and ranges, but they also have characteristic rates of growth and growth patterns. Kind of climate, I talked about climate as well. Changes in climate because humans are affecting the levels of carbon dioxide, and that's affecting how much heat is retained. Lots of evidence for this, just like for other theories. And so we have lots of different multiple lines of evidence all pointing in the same direction. Among the best evidence are big things like Arctic sea ice. We can see that. We have nice physical evidence of that disappearing. Another really nice evidence of long-term climate change is shrinking glaciers. We've had these things marked and we know their extent. We have, again, aerial photos. We have physical ground truth for that as well. We've got records of those historic records that are quite nice. And then sea level rise, nice things. Again, you know, hard to fake stuff like this. They don't talk about this when they talk about fake news because you can't really fake it. Okay, so, there's my population stuff. Somehow climate change got moved into the wrong thing. Okay, so you have the exponential and logistic growth. In logistic growth, there's some sort of limiting factor that slows growth rates. Okay, in organisms that are exponential growth are often limited by kind of disaster type things that are density independent. like weather events or things like that. And so they have what's called an opportunistic life history. These are like insects. They're just going to keep reproducing really fast rate until the weather gets cold and they all die. Other organisms like elephants are more equilibrium life history. They reproduce very slowly. It takes them 15 to 20 years to mature. And so they don't even start reproducing until they've been around for 20 years. They've got to grow and learn and things like that. Then they have one offspring at a time, which they take care of for years and years and years. And they can, even under bad conditions, they can reproduce because they can adjust their rate of reproduction, adjust growth rates and things like that. And of course, that's us. Okay. And so we're growing at exponential growth though, but that growth rate is starting to slow. How fast it slowed depends on how, will determine how many people the Earth ultimately gets, but we are showing slowing growth. We're somewhere like right here where the growth rate is actually dropping. So we don't have a stable population yet, but here this shows birth rates, and birth rates are falling. Okay, we're 2020, so we're somewhere like right there. Okay, and so this is as death rates are falling throughout the world, birth rates are falling. Okay, so I'll stop the sharing there. Does anybody have any questions? Did I go over the essay questions? Yes? No?
Yeah, you did.
Okay, so I don't have to do that. We've only got a couple minutes left. The I opened up the key to the practice exam just before I started here. So that practice exam is open. You can look at that. Again, I want to remind you that the exam will open on Monday. There'll be somewhere between, it'll be less than 100 questions, probably 70 to 80 questions, multiple choice, true, false, and then one essay to write. Okay, those essays are there. You'll be randomly assigned one or the other. So you'll be there. You'll have 2 hours to do the exam, only one chance. If you would have an internet problem, you would text me immediately. I think my understanding of how it works, like say your Wi-Fi would go out momentarily, is that it would let you re-log in if you re-logged in within the next 2 hours. Because I've had several people try that. Otherwise, if you re-logged or if you couldn't get back in, then you have to get in contact with me and we've got to figure something out. So I would look at what you did and then do something there. So I'll be watching my email for the week, and I would try and get back to you relatively soon. Okay, so I'm going to stop unless people have any kind of questions. Does anybody have any questions?
Like, what do you suggest we study the most? Because like you said, we don't have time to read everything, so
Right.
what should we focus on?
I would look at my old exams. Okay, I would look at the old quizzes. I think that's really helpful. All the quizzes are open, so you can, if there's a quiz that you're not sure that or that you missed, you can go take it, and so that would be very helpful so that you see those questions. Let's see. I would look at chapter summaries. That would be a good thing. I would look at vocabulary lists. If you made any kind of review materials for the previous exams, I would look at those. The essay questions are both kind of on ecology. There will be a little bit more ecology questions on the final, but it won't be half ecology. It'll be, there'll be, the essay will be on ecology, and then maybe 10 questions. So look over old exams, look over review materials for old exams, look over old vocabulary. I'm not going to surprise you with a whole bunch of new questions. There won't be there won't be a huge number of new questions, or if there are new questions, they will ask about the same things that the old questions asked about. So, okay. Good. That's very good. Very good. Good to ask about. Thank you. Anyone else? Okay. All right. I'm going to stop and end the meeting, and I will try and, I think I've recorded this, so I will try and post this thing on the Moodle page. It takes a long time to process this because it's long, so it'll take me an hour or two. So, okay, thank you. Just study like you studied from before, and things should be okay. You know, we're going to get through this. We're almost done. And so, you know, if you have any questions, email me. I'm not at the office too much, but I check the email several times every day. Okay. All right. Thank you. Good luck.